Hi, I'm Michael Wong from the Department of Neurology in the Department of Molecular and Integrative Physiology at the University of Michigan. I'm also on the staff at the VA Ann Arbor Healthcare System. I'm a physician scientist with training in vascular neurology, and my laboratory has been studying the pathogenesis of inherited vascular dementia for nearly 2 decades. Today I'll be giving you an overview of vascular dementia, one part of a series of web-based sessions organized by the Michigan Alzheimer's Disease Center for Scientists. Our hope is to convey core information about neurodegenerative diseases to provide you with a foundation for deeper learning about important neurological disorders. These are my disclosures, and these are the key terms that we'll be covering today. Vascular dementia is a topic of high importance to public health in an era of prolonged longevity and increased incidence of age-related diseases. In many ways, vascular dementia represents a dualistic disorder. As we shall see, it is an old disorder and a young disorder at the same time. It affects large geographical areas of the brain and small microscopic areas at the same time. And it comes in common sporadic forms and in rare genetic presentations. This breadth makes for a difficult topic to discuss in a single session, so I'll be limiting our discussion today to simply covering 2 core topics listed here. The purpose here is not to be comprehensive, but to provide you with an opportunity to expand future knowledge, which can be gleaned through many excellent reviews in the literature. In brief, the objectives of this discussion are to 1. Define vascular dementia, its impact, and its clinical diagnosis, and then 2. Describe the heterogeneity of this disease by illustrating several of the vascular conditions that lead to dementia. So how do we define vascular dementia? There's both a short, simple definition shown here and a more complicated definition that we'll discuss later. This simple definition, dementia caused by vascular disease of the brain, seems straightforward, but as we shall see, this area is quite complex. Historically, it is interesting to note that vascular dementia has been recognized for over 3 centuries. The famed Thomas Willis, the same physician who described the collateral circulation at the base of the brain, cited 6 causes of dementia in his treatise, Practice of Physic, in 1684. The 3rd cause of dementia he described was, quote, great strokes or bruising of the head. For its time, this was a keen acknowledgement that vascular disease could affect long-term brain function. 200 years after Willis, another notable name in neurology described vascular dementia by careful pathological examination of brains of dementia patients. This famed neurologist published the paper Arteriosclerotic Atrophy of the Brain in 1894, which described detailed histological abnormalities of vessels of the brain in a subset of dementia patients in Germany. It turns out that Alois Alzheimer recognized vascular dementia a decade before his description of his most famous form of dementia, which was named after him, Alzheimer's disease. Alzheimer, in fact, described 3 main categories of dementia. In the first category, he conjectured a vascular cause of dementia based on arteriosclerotic pathology in brains of demented patients, which I've illustrated with an immunohistochemical image of stiff, protein-filled arteries. The second category, which he felt was less common, was a senile dementia that later he found to feature plaques and tangles, the modern hallmarks of Alzheimer's disease. Third, also very common at the time, was dementia due to syphilis. Interestingly, during the early part of the 20th century, the arteriosclerotic view of dementia took hold of neurology and was considered the major cause of dementia. It wasn't until the later part of the 20th century that Alzheimer's disease captured the interest of the neurological community, and it was discovered in pathological samples that Alzheimer's disease makes up a great fraction of dementia cases. So from the 1960s to the beginning of the 21st 21st century, the pendulum swung in the direction of senile plaques and tangles, resulting in an overwhelming emphasis on Alzheimer's disease. At the same time, vascular causes of dementia seem to recede into the background. But historical views about dementia have now come full circle. The modern thinking is that dementia is largely a multifaceted disease entity with complex pathologies. The current thinking is that neurodegenerative processes are often mixed, with the most common mixture composed of two entities: Alzheimer's disease pathology, plaques and tangles, and vascular pathology. There is a major initiative to investigate vascular dementia from the top levels of the US healthcare system. NIH-sponsored groups have cited vascular dementia as potentially the most important factor influencing brain health in the elderly. In 2014, recognition of the importance and prevalence of vascular disease in health was the focus of a workshop on the NIH campus in Bethesda. Since then, there has been a continued emphasis on vascular contributions to dementia at the federal level. The big picture numbers behind the epidemiology of vascular diseases of the brain are hard to contest. Although methodologies differ in the calculation of vascular dementia prevalence, the vast majority of studies agree that vascular dementia is the second most common cause of dementia, following only Alzheimer's disease in its prevalence. Placed in terms of rates of disease, vascular dementia follows very closely behind Alzheimer's disease, with a current rate of 14.6 diagnoses of vascular dementia in every 1,000 person years. Many studies have shown that vascular dementia is the second most common cause of dementia, following Alzheimer's disease. Vascular dementia prevalence exceeds the sum of all other causes of dementia, excluding Alzheimer's disease. Paramount to the accurate calculation of impact of vascular disease is the proper definition of vascular dementia. As we mentioned before, at first approximation, vascular dementia is easy to define. It is dementia caused by vascular disease. But it turns out that this is quite a nebulous definition that is difficult to ascertain without imaging or pathology. This has spurred, with good reason, many efforts to formalize definitions that can be applied clinically and for research purposes. So, how do we make the diagnosis in a patient we are seeing in the clinic? The first effort to define vascular dementia was by Vladimir Hachinsky from the University of Western Ontario in London, Canada. Dr. Hachinsky, a stroke neurologist, was a very early proponent of vascular dementia in the 20th century and enabled vascular dementia to walk on its own and emerge from the shadow of Alzheimer's disease. The Hachinsky ischemia score was one of the first scoring systems used in neurology and the first popular tool used to estimate the probability of vascular dementia. The Hachinsky score holds great appeal for its numerical nature, ease of use, and clinical efficiency. A very important feature of the score are categories that weigh ischemic stroke signs and emphasize the staccato tempo of progression felt to characterize vascular dementia. However, limitations have been noted. It's generally thought that the scale is less specific than later criteria and oversimplifies the vast diversity of vascular dementia. In addition, as an early measure, the Hachinsky score does not incorporate contemporary clinical information such as MRI. A joint collaboration between American and French investigators resulted in the next major advance in formalizing clinical criteria for vascular dementia. The so-called NINDS-ARIN criteria included refinements to Hachinsky's formulation, requiring causative links between dementia and cardiovascular disease, and utilizing clinical features supportive of vascular disease. In addition, these criteria accounted for atypical presentations and encouraged the utilization of histological and radiological criteria. Additional criteria for the diagnosis of vascular dementia have also been proposed by the State of California Alzheimer's Disease Diagnostic and Treatment Centers. These criteria, like the NINDS-ARIN criteria, were drawn up by a consortium of clinical researchers and incorporated radiological data and more contemporary clinical data. All of these detailed criteria have been published and are widely available. Finally, the DSM-5 recognized vascular dementia as a diagnosis, and vetted consensus criteria have been established. This is perhaps the most updated set of criteria that incorporates all clinical data, such as MRI evidence. In addition, more recent genetic information, such as sequence analysis for CADASIL, is discussed. In summary, since the very beginnings of interest in vascular dementia, the definitions have undergone an evolution. The core features of vascular dementia, such as focality of neurological findings and sudden nature of symptoms, are important clues, but new criteria have been added to match advanced imaging available to the clinician. It is projected that future updates to these criteria will add more detailed molecular data and add the use of biomarkers that become available in the future. So, returning to the simple definition of vascular dementia, we now have several more detailed systems that can be used for diagnosis. Sudden and focal clinical symptoms and brain imaging are critical. But the advent of imaging has led to recognition that vascular dementia is actually strikingly heterogeneous, which is the emphasis of the second half of our discussion. MRI imaging has revealed that vascular disease encompasses a large variety of processes and consequences, all of which may converge to cause dementia. The wide spectrum of processes that result from vascular disease is illustrated here in 3 different MRI images. The variability of vascular lesions results in a mixture of brain changes. These changes can be major in the case of large territorial strokes, shown on the left. When vascular disease affects penetrating vessels of the brain, it may cause gradual and insidious changes in the case of small vessel ischemia, which affects deep white matter, shown in the center. Third, vessels can rupture, causing hemorrhages, which can be either large or small and affect cortical or deep structures. Adding to this complexity, the variation in territories that are affected by vascular disease, ranging from anterior to the posterior poles of the brain. Damage to different areas can cause different symptoms, depending on location. Due to the complexity of vascular disease, it is not surprising that specific presentations of vascular dementia vary. Though dementia is generally defined as involving 2 or more cognitive domains, in vascular dementia, these domains can vary. As such, the typical domains that are involved in Alzheimer's disease, memory and language problems, are not consistently present or absent in vascular dementia. What is noted in a large number of cases, however, is a temporal relationship between slippage in cognitive domains and a stroke within 3 months, although this is variable as well. I'd also like to point out that vascular dementia is complex because it coexists with other disorders. What cannot be ignored in any discussion of vascular dementia is the overlap between Alzheimer's disease and vascular disease. Both of these pathologies are intertwined at many levels, as represented in this Venn diagram. Because vascular dementia and Alzheimer's disease are related, separating them clinically and in research protocols presents challenges. This complexity is seen at multiple levels. First, Alzheimer's disease and vascular dementia share risk factors. This promotes the co-occurrence of both vascular dementia and Alzheimer's disease in the same population and even in the same person. This is made apparent in pathological examinations of brains, which show the evidence of vascular dementia and Alzheimer's disease in the same individuals. Another complexity of these diseases is that vascular diseases serve as stimulants to Alzheimer's disease. In other words, vascular disease increases the probability of developing Alzheimer's disease. And the presence of vascular disease significantly increases the speed of progression of Alzheimer's disease. In sum, this results in a complex pathology. Diagrammatically, the current thinking is that Alzheimer's pathology may promote vascular disease. and that vascular disease likely promotes the pathology of Alzheimer's disease. This positive feedback interaction makes it likely that vascular disease and Alzheimer's disease coexist in the same individuals. As we shall discuss a little later, there are direct biochemical interactions in Alzheimer's disease that affect the vascular system, the most widely recognized being deposition of amyloid in vessels, known as amyloid angiopathy. This coexistence and feedback between vascular dementia and Alzheimer's disease can be represented here, a depiction of mixed dementia in which features of both vascular dementia and Alzheimer's disease occur in the same brain. We are not very good at definitely separating mixed disorders from pure Alzheimer's disease and vascular dementia. This operationally complicates the clinical and research definitions of vascular dementia. Among the three vascular pathologies we see often in the clinic, one in particular has been the focus of a great deal of recent interest at the epidemiological, clinical, and basic scientific level: small vessel disease of the brain, SVD. SVD involves changes of vessels that measure less than 500 microns in diameter. The characteristic features of small vessel disease are shown here in this immunohistochemical analysis of a diseased vessel stained for collagen. Vessels become stiff and their walls undergo fibrotic changes. The microscopic changes we see include vascular smooth muscle cell loss, protein deposition, and wall thickening. The significance of SVD cannot be underestimated. A series of studies of different populations around the world have now estimated SVD, detected by white matter hyperintensities on MRI, as a strong risk factor for dementia. This is illustrated in the meta-analysis from 2010 by Debet and Marcus, which demonstrated that the risk of dementia in the presence of SVD increases by 100%. The importance of SVD is amplified by a number of other features. As more and more of the elderly have MRIs of the brain, it is apparent that large fractions of the elderly population have SVD. Some estimates are that over 65% of people over the age of 65 have MRI evidence of SVD. In addition, SVD is a risk factor for both vascular dementia and Alzheimer's disease. In total, SVD may be an important subset of vascular pathologies that drives vascular dementia and mixed dementias. We can learn a great deal from inherited monogenic forms of disease. The value of these entities is that they are relatively pure diseases, in contrast to vascular disease of the elderly that is often admixed with mixed dementia. The best characterized SVDs that are inherited include CADASIL, CARASIL, and the collagen 4A disorders. Each of these diseases is caused by mutations in a single gene. The most common of these by far is CADASIL, which we will spend some time discussing since it provides clinical and scientific insight into vascular dementia. Although not common, CADASIL occupies a special place in the world of vascular dementia. As mentioned, a subset of vascular dementia is linked to SVD pathology. Within this set of SVD patients, a small percentage of patients have inherited disease. Finally, among cases of inherited SVD, the majority of these are CADASIL. What is CADASIL? This acronym stands for Cerebral Autosomal Dominant Arteriopathy with subcortical infarcts and leukoencephalopathy. As the name suggests, this is a brain disorder inherited in an autosomal dominant fashion that features pathological changes in cerebral arteries. The ultimate cause of subcortical infarcts and diffuse white matter changes that are demonstrated in this MRI image. The arteries of the brain that fail exhibit some very characteristic changes that are seen here and in the next slide. There is thickening of arteries, especially of the white matter, with fibrotic changes that involve the full thickness of the vessel wall. In addition, we observe loss of normal smooth muscle cells and increased collagen content. The collagen accumulation stiffens vessels of all calibers and permeates through the full thickness of the vessels. Also of note, there is accumulation of Notch3 protein, a transmembrane signaling molecule that is mutated in CADASIL within the medial layer of arteries. The focal accumulation of Notch3 in these regions of the vessel that feature degenerating smooth muscle cells is characteristic of CADASIL. Thus, CADASIL appears to be associated strongly with degeneration of specific cell types in the arteries of the brain. We mentioned that Notch3 protein accumulates in CADASIL vessels. Under electron microscopy, one can also observe punctate accumulations of protein known as granular osmophilic material, GOM, that are pinpoint aggregates that abut the plasma membrane of smooth muscle cells. These deposits are pathognomonic for CADASIL and could represent accumulations of misfolded proteins, including Notch3. These features of CADASIL pathology suggest that the disease could result from or be associated with abnormal protein conformations and could indicate proteinopathy, a frequent feature of neurodegenerative disorders. To summarize, CADASIL is thought to cause vascular dementia through degeneration of the smooth muscle cells in arteries within the brain. These changes in cell content are linked to protein accumulation, possibly protein aggregation, and vessel failure that results in diseases of the white matter. Ultimately, this contributes to vascular dementia. The genetic basis of CADASIL was revealed in 1997 by Joutel and colleagues after an exhaustive analysis of French families, gene mapping, and sequencing. CADASIL was determined to be caused by mutations in the Notch3 protein. An examination of the Notch3 mutations in CADASIL provides several insights into possible molecular mechanisms of disease. As we mentioned, Notch3 protein accumulates in CADASIL vessels in the smooth muscle layer around degenerating cells. The mutation appears in a part of the gene that encodes the extracellular EGF-like repeats, which are peptide motifs that contain 6 disulfide bonded cysteines. Very interestingly, virtually all the mutations that cause CADASIL include cysteine residues. The mutations either trigger a loss of a conserved cysteine or a gain of a new cysteine. Since cysteine is likely critically involved in the structural stability of Notch3, these genetic findings provide compelling support for protein misfolding as a mechanism in CADASIL. Returning to the theme we have been stressing, vascular dementia is heterogeneous. Even the pure vascular dementia CADASIL is diverse. In the same family, where all affected individuals have the same mutation, there are vastly different clinical courses. For example, some CADASIL patients can be cognitively normal, while others in the family have a significantly accelerated dementia. Other patients have recurrent strokes, while others remain stroke-free. Life expectancy and progression rates diverge dramatically as well. These observations on the diversity of a small set of vascular dementia patients illustrate some of the challenges in the diagnosis and research on vascular dementia in general. To wrap up our short discussion of CADASIL, I would like to point out a few parallels between this disease and other more widely recognized degenerative disorders. CADASIL is likely a disease that features protein conformational changes of Notch3. This is similar to other diseases of the brain that each feature conformational alterations of a unique protein listed here. To further extend our analogy to other neurodegenerative disorders, CADASIL is similar to other diseases in that it targets a specific cell population within the brain. In CADASIL, the vascular smooth muscle cell is marked for cell death and neurodegeneration, much like specific cells are targeted in Alzheimer's disease, CJD, and Parkinson's disease, as shown here. To summarize, there is reasonable evidence that the core mechanisms of classical neurodegenerative disease may be shared in CADASIL. It remains to be seen whether other forms of vascular dementia share molecular and cellular disease mechanisms with these canonical neurodegenerative disorders. A final vascular pathology for discussion today is hemorrhage. Any compromise of vascular integrity can lead to bleeding in the brain. Hemorrhage can predate and serve as a trigger for dementia in the future. For example, large hemorrhage due to hypertension is a risk factor for dementia. Recurrent hemorrhage of larger vessels also sets the stage for chronic stepwise impairment. Alternatively, hemorrhage can be symptomatic of a pathological process that causes dementia. To exemplify the chicken and egg nature of vascular disease, let's discuss a process called amyloid angiopathy. Cerebral amyloid angiopathy, CAA, is a process that targets the small and medium-sized cerebral vessels, with a preference for structures of the gray matter. Vessel walls are compromised by deposition of amyloid protein, most commonly a beta peptide featured in Alzheimer's disease. Here we see a leptomeningeal artery stained for amyloid protein in red. The protein deposition results in smooth muscle cell loss and impaired reactivity and wall integrity. A majority of Alzheimer's disease patients has some degree of amyloid angiopathy. In some instances, amyloid angiopathy occurs independently from Alzheimer's disease. In general, a patient with symptomatic CAA, the primary manifestations are hemorrhage in the brain that frequently is linked to cognitive deterioration. When larger arteries rupture, cortical bleeds can become apparent. Meanwhile, involvement of small vessels of the brain leads to small and sometimes numerous hemorrhages that can be detected on MRI. This microhemorrhagic state is illustrated in this SWI image, which shows innumerable round dark areas in the cortex, largely sparing the white matter. It is worthwhile re-emphasizing that CAA is very strongly linked to Alzheimer's disease epidemiologically and biochemically. The presence of CAA is a marker for more aggressive Alzheimer's disease, which is consistent with the known effects of vascular disease on cognitive deterioration. On the other hand, the inability of the brain to handle amyloid protein is likely to contribute to vascular disease. The overall message is that CAA rears its head in many ways and is also coexistent with other pathological conditions, again illustrating the complexity of age-related vascular disorders. This is the very last part of this presentation, and I would like to now circle back to vascular dementia and talk a little bit about treatments before wrapping up. We are in the early stages of studying vascular dementia. Although vascular dementia is an old disease, we are in a new phase of knowledge gathering about mechanism and pathology. Thus, it is not surprising that we do not yet have effective therapies. There are no known direct therapies for vascular dementia. However, there is a good body of literature that describes approaches to stabilizing the brain vasculature. This includes reasonable low-risk measures that depend on the characteristics of each vascular dementia patient. For example, it is reasonable to treat a vascular dementia patient who has had ischemic stroke with aspirin and high-intensity statins. Similarly, it is reasonable to optimize blood pressure control in patients with hypertension. Smoking cessation should be encouraged to reduce vascular risk. It is known in studies of CADASIL patients that ischemic stroke is directly related to cigarette smoking and to hypertension. A modest diet and exercise program can be useful in vascular dementia patients, though this has not been studied in detail. So where do we go from here? I've already mentioned some of the gaps in knowledge about vascular dementia. We have discussed the difficulty in establishing a diagnosis and the high variability of disease. Experts agree that a biomarker for vascular dementia could be an important tool for both diagnostic and research efforts. And in addition, establishing molecular mechanisms of disease will be very important in the generation of therapeutic targets. This is our third to last slide. I'd like to leave you with some specific information that is useful in thinking about vascular dementia. Vascular dementia is the number 2 dementia after Alzheimer's disease. Vascular dementia is defined by specific criteria, but is very heterogeneous. Small vessel disease pathology causes an important subset of vascular dementia. And finally, CADASIL is a major inherited form of small vessel disease. I hope you will agree that vascular dementia is an evolving field of major impact, and that we will almost certainly refine our ability to accurately diagnose vascular dementia in the future. This will be a very important step in taking us beyond supportive and symptomatic care for this challenging problem. At this point, I'd like to thank you very much for joining me, and thank you for your interest in the Michigan Alzheimer's Disease Center.